Vitamin B1, also known as thiamine, is one of around 10 B vitamins that are necessary for our biological processes to actually function correctly. It is an energy-producing vitamin, meaning that somewhere in its main use inside of the human body, it is used to convert macronutrients, calorific macronutrients, into acetyl-CoA, and then henceforth into the Krebs cycle and into oxidative phosphorylation and the electron transport chain. Somewhere along the line, these B vitamins will be used in order to help this process continue. And vitamin B1 has a couple of specific uses inside of this pathway. Now, all the B vitamins are water-soluble vitamins, so in terms of sort of dietary intake and stuff like that, the B vitamins in general are quite often the ones that you are most likely to be deficient in. This is because very little amounts can be stored inside of the liver, and when it's inside your bloodstream, it can very easily just sort of go out the kidneys and then be excreted in your urine. Like, it's not all going to stay inside your bloodstream. It is also not all going to go out, but it is going to gradually sort of decrease and decrease and decrease. It's not able to be stored in your body, like all of it, and all be used. So there are certain foods high in vitamin B1, legumes and whole grains, and some leafy vegetables. But vitamin B1 deficiency, if you don't get enough vitamin B1, is something known as beriberi, where you have a lot of muscle spasms and muscle fatigue. A lot of it's to do with muscles because it is directly involved in energy production. And that is because it plays a major role in an enzyme known as pyruvate dehydrogenase. Now, thiamine, before it can even sort of react and engage with this enzyme, you have to eat it first in certain foods such as legumes and whole grains. It will then be absorbed into the bloodstream by the small intestine, and then it will be converted in cells into its active form, which is known as thiamine pyrophosphate. And this reaction, thiamine to thiamine pyrophosphate, is catalyzed by the enzyme thiamine pyrophosphate kinase. Now, this means that thiamine has two phosphate groups on it, and thiamine as a molecule itself, it's already a bit of an interesting one, but not too complex. It has a five-membered ring with an N+ charge, which is somewhat rare, as well as a sulfur, which is actually quite rare inside of a lot of molecules. Now, I'm not sure, there's going to be a specific part on the TPP, which is actually able to mean that it can bind to certain parts of the whole pyruvate dehydrogenase complex and pyruvate itself. And that is important. I'll probably circle the little things on the images that I've got on screen. But the reason that this enzymatic reaction is so important is because this converts pyruvate into acetyl-CoA and is essentially the bridge between glycolysis and some amino acid metabolism to the Krebs cycle, which starts with acetyl-CoA as well as oxaloacetate converting into citrate. But this reaction is also non-reversible. So TPP, because it is a cofactor in this process, it cannot convert acetyl-CoA back into pyruvate. That is just not able to happen biologically. But what this whole enzymatic complex does is TPP will covalently bind to the active site of the enzyme, and then TPP will covalently bind to the pyruvate, but it won't covalently bind to, maybe, for instance, you would have seen the pyruvate molecule, it's got like an O- charge. That doesn't mean that it's going to like ionically bind to the N+ charge in the thiamine. What's actually going to happen is, in this reaction, CO2 is excreted in the end. If you look at the sort of whole basic reaction, CO2 comes out as well as acetyl, and the whole CO2 being excreted is because TPP will bind one carbon in to the pyruvate molecule, and there is a COO that's just going to be left. This creates a molecule called hydroxyethyl TTP. This will then be transferred to a lipoamide, which must just be a fat with some kind of amide group on it. I'm not exactly sure of the specifics on that. And that lipoamide will then transfer the acetyl group onto a coenzyme A molecule. Now, I've done an entire video on how coenzyme A specifically is made, and you can check that out if you want. And that's basically the whole mechanism of how TPP actually transfers pyruvate to acetyl-CoA. Now, there's probably a bit more detail, admittedly, to be had in this kind of process, but it was hard to find a lot of clear-cut things that I could synthesize and put easily into a video to be able to tell you guys that. But there are other functions of TPP, including being a cofactor of an enzyme as part of the non-oxidative pentose phosphate pathway. Now, the way that TPP helps out the enzyme transketolase it's actually quite complicated. I made a video on xylitol metabolism, xylitol will turn into xylulose 5-phosphate, and xylulose 5-phosphate will eventually turn into ribose 5-phosphate. But that's actually quite a complicated process. It was quite confusing because you ended up getting like 7-membered sort of sugars, which was incredibly interesting, and there had to be sort of 2 carbon units randomly transferred between different molecules, because there was glyceraldehyde was a 3-carbon sugar, and so I think it was something like 5 would be reacted with 3 to form like 7, and etc. But it was very complicated. In the end, 3 xylitol molecules get converted into 2 fructose 6-phosphate molecules, as well as 1 glyceraldehyde, and that involves the transfer of carbon units. Some of these transfers included 2 carbon units. Sometimes 2 carbon units would be transferred to 3 carbon units. In this case, it's xylulose and ribose having the 2 carbon units sort of transferred off each other and then back onto each other. Hydroxyethyl TTP is part of this process, because the hydroxyethyl part of TTP, the whole acetyl-CoA sort of structure that is attached to this molecule, it's the exact same structure as the 2 carbon unit that is taken off. So I remember what's interesting is when I was editing this video, there was no specification as to what exactly that molecule was, but now I know that it is this thing that's actually attached to TTP, the coenzyme, rather than just something floating around in sort of the enzyme's active site. It's actually binding to something, and it's able to be transferred stably rather than just sort of happening at a weird rapid rate without any real control, in a weird sort of way, because I guess maybe that's the way that I played it off or acted like that was what happened, but that's not the case. It's actually much more controlled, and TPP, vitamin B1, is the reason that reaction is able to exist. And if these reactions don't exist, as I've said at the start of the video, muscle fatigue and not the most optimal coordination with spasms and stuff like that means that all these things occur if you don't have an adequate vitamin B1 intake. But with that, I hope you enjoyed, and I recommend subscribing for more content like this, and have a good day.